Hello everyone, thank you for coming to my talk. Today I'm going to share with you some of the interesting findings from my lab in the past year. This work is mainly led by Anderson along with Brock Juliker, a productive undergraduate team. We also established an international collaboration with modelist Professor Che from Cheung Kong University. So this talk will not only demonstrate experimental findings, but also include insight from theoretical perspectives. The topic of this talk focuses on flow coupling between active fluid and passive fluid. This movie shows extensile microtubule bundles, which is an active fluid in our system, self-organized into a circulatory flow when confined in a water in oil droplet. We are going to show how we can trigger or suppress the circulation without touching the droplet, just like Jedi uses the Force to manipulate the objects that are out of reach. Before that, please allow me to briefly introduce my lab. My lab locates in Life Sciences and Bioengineering Center. It is a resourceful facility along with research groups from multiple disciplines such as physics, biology, and chemistry, allowing many PIs to interact and break the boundaries among different labs. This is my lab. Established since 2017, sitting at one triangular corner of the LSBC floor with window walls and ample space. In the past years, my lab has recruited 12 students. Currently, we have 7 active students working on various active matter projects from fundamental study to technological applications and K-12 outreach activities. We also collaborate with various universities such as Brown University, Michigan State University, and Cheung Kong University. We are also the IRG2 contributor in Brandeis MerSec, which is an NSF-funded research center where we can exchange our research progress and receive feedback. Another great news is that we recently received a 5-year NSF Career Award, allowing us to bring our research and education activities to the next level. We are looking forward to the new challenge and new adventures in the following years. Now let's begin. Before I talk about flow coupling between active and passive fluids, let's first introduce active fluid. Active fluid is part of an active matter. But what is active matter? Well, according to its name, active matter is a material that is active and mobile. Based on the second law of thermodynamics, to maintain the activity, the material must constantly consume fuels. Since the fuels are constantly consumed, entropy continuously increases. So, the system is far from equilibrium. Because of these characteristics, active matter is fundamentally different from passive matter. Here is one example. In its movies, we see colloidal particles suspended in water. Water molecules randomly and constantly bump onto colloids, causing them to jiggle. In my lab, Taegan video recorded the colloidal jiggling, but he only recorded them once. So these two movies are the same movie, except one is played forward, the other is played backward. Can you tell which one is played forward and which one is played backward? Very hard, isn't it? The answer is the left one is played forward and the right one is played backward. However, without me telling you which is which, it's nearly impossible to tell, right? Why? The reason you cannot tell is because the movies I showed you is a system in equilibrium. An equilibrium system has no time. Going forward and backward follow the same equation. So fundamentally, forward and backward have the same physics. Now let's look at another example, sperms swimming. Similarly, in these movies, one is played forward, the other is backward. Can you tell which is which now? Probably yes. Why can you tell in this case? The reason you can distinguish forward and backward is because in this system, sperms are swimming, constantly consuming biological fuels to beat their flagella. The system is far from equilibrium. The second law of thermodynamics says that this system will increase its entropy over time. And since entropy increases with time, we can tell the direction of time. So from this example, we know that active matter is different from passive matter from a fundamental perspective of time. Passive matter has no concept of time. Active matter has a time arrow. Now we have learned about active matter. We can start recognizing the active matter in our daily life. Basically, the material that can move, consume fuels, stay far from equilibrium is categorized as an active matter. As such, active matter can be ants, bacteria, sperms, bats, cells, and even humans are active matter because we walk, we eat, and increase the entropy of the world. However, active matter is not only limited to living things. Scientists have also invented various active matter systems where the constituents can move by itself. Examples are colloidal servers, Quincke rollers, gliding actins, micromagnetic rollers, and genus particles. My postdoc advisor even created this squirming object which looks like a living thing, except it is not. Since active matter can move by itself, scientists also wonder whether they can regulate their motion into a spontaneous coherent flow. And they succeeded. The common trick is to confine active matter in circular confinement. Scientists found that when active matter is confined in a proper circular chamber, they self-organize into a circulatory coherent flow. This phenomenon is ubiquitous and has found in many active matter systems ranging from insects, bacteria, cells, grains, colloids, and even microtubules. In this talk, we are going to focus on spontaneous circulation of our active matter. Our active matter system contains three major components. The first component is microtubules. We extracted microtubules from animals and we suspended them in our test tubes. The second component is depleting polymers. These polymers bump onto pairs of microtubules, create osmotic pressure, or called depletion, to bundle microtubules. The bundled microtubules will be breached by our third component, kinesin motor dimers. These motor dimers slide pairs of anti-parallel microtubules apart, causing interfilament sliding. In the lab, we saw such interfilament sliding. In this video, the polarities of microtubules are colored. Then we can see that when microtubules were parallel, nothing happened. But when they were anti-parallel, they slid apart immediately. Such interfilament sliding, along with the depletion force, collectively cause microtubule bundles to repeatedly extend, buckle, and anneal. These repeated dynamics accumulate to millimeter scale, cause microtubule bundles to form a percolating network whose structure constantly rearranges. This self-rearranging structure looks like a gel, so we call it active gel. In my postdoc study, I found that if we confine this active gel in the toroidal boundary, then we can regulate the gel motion into a unidirectional coherent flow. The confinement is not limited to toroids, it can also be a cylinder which can support the circulatory flow. In this movie, we dope tracers to track the solvent flow, allowing us to better characterize the flows quantitatively, such as analyzing flow profile and mean speed. This circulatory flow is the consequence of interactions between active fluid and confinement surfaces. Confinement surface causes the microtubule bundles to align and form a distorted nematic layer. This layer extends because of extensile microtubule bundles, which apply a force along the surface to induce pipe flows. As such, the knowledge of how active fluid interacts with boundary is the key to regulate active fluid flows, as well as direct active fluid to perform mechanical work for downstream applications. Boundaries contain two characteristics: shape and surface property. For shape, one of my graduate students, Edward Jarvis, is working on exploring self-organization of active fluid confined in various geometries such as trapezoids and triangular prisms. Then, how about surface property? In this case, the surface enforces a no-slip boundary condition, which sets a zero flow speed at the boundary. What if the boundary is instead slippery, or more specifically, fluid? Will the fluid confinement still support circulatory flow? Answering this question requires an experiment that confines active fluid in a fluid boundary condition. To create a fluid boundary condition, we confine active fluid in a water-in-oil droplet that were compressed between a pair of parallel plates. Since water and oil are both fluid, the interface is fluid. So this intra-droplet active fluid is effectively confined in a fluid boundary. However, in this movie, we learned that this active droplet is motile because of its interior active fluid activity. Its motility will prevent us from long-term observation on the intra-droplet active fluid flows. To immobilize the droplet, we created a surface tension-induced potential well in the following way. Since the droplet was compressed between a pair of parallel plates, the droplet will not cause any energy if moving horizontally. However, if we slightly curve the ceiling, as well as drill a thin well, then moving away from the well will cause surface energy. As such, droplet compressed in our dome well ceiling will become immobile. Within this immobilized droplet, active fluid develops a circulatory flow, which indicates that circulatory flows can be supported even when the boundary is fluid. However, fluid boundary and solid boundary are fundamentally different. Solid boundaries isolate the fluid dynamics within the boundaries. Dynamics outside the boundaries play no role in the dynamics inside the boundaries. This is different from fluid boundaries. Since fluid boundaries is fluid, the fluid inside the boundary can interact with the fluid outside the boundary. Therefore, how such interactions affect the formation of the circulatory flow is the focus of this talk. To investigate such an interaction, we characterize the intra-droplet active fluid flow by doping the fluid with tracers. we can track the tracer motion to calculate the average azimuthal velocity as a function of time. In this case, the droplet flowed at approximately 10 micrometers per second for hours. Then, to characterize how well the fluid circulates, we defined a circulation order parameter, which is a mean fraction of azimuthal velocity. The order parameter is 1 for perfect circulation and 0 for random flows. In this case, the circulation order parameter remains at approximately 0.6 for hours. With these analysis tools, we are able to characterize how the fluid boundary affects intra-droplet circulations. Our immediate finding was that the circulation depends on the boundary geometries. When the radius of droplet was increased, the circulatory flow was suppressed. To characterize the role of droplet radius on the formation of a circulatory flow, we systematically varied the droplet radius in a fixed milli-fluidic device and measured circulation order parameter. Our measurements showed that the circulatory flows depended on the droplet radius. Circulation is only supported in the droplets with a specific radii. However, when we performed the measurements again, we got different results. Why would the results be different? After careful checks, we found that the results were different because the experiments were different. While we measured the circulation order parameter within the same set of droplets, they were immersed in a different milli-fluid device. The one has a container radius of 5 mm, the other is 3.5 mm. Here, I would like to remind you that this result would never happen in solid boundary system because the fluid inside the solid boundary would never care about the thickness of the boundary. This is the consequence of fluid boundary. From this observation, we hypothesized that active fluid flows within the droplets are not only affected by droplet geometries, but also by container geometries. To verify our hypothesis, we performed two experiments. The one has a droplet that supports the circulatory flow. The other has a droplet of the same shape, but the droplet was loaded in a smaller container. In this droplet, we found that the circulation did not develop. This result indicates that even though these two droplets have the same shape, loading them in different sized containers that hold different amounts of oil influences active fluid flows differently. To characterize how the oil configuration affects active fluid flows, we designed another set of experiments, where we measured circulation order parameter of active fluid within droplets of the same shape while systematically varying the oil layer thickness, and then plotted the circulation order parameter as a function of oil layer thickness. Our measurements showed that oil layer thickness indeed influenced the formation of intra-droplet circulatory flows. This influence is prominent when the oil layer is thin. For a thick oil layer, the circulatory flow is nearly unaffected. To verify such a characteristic of influence is not limited to droplets of specific shapes, we repeated the measurements with different sizes of droplets. In this case, we kept the droplet radius the same but reduced its height. In this droplet, we observed similar characteristic. When the oil layer thickness was small, changing the thickness significantly altered the circulation order parameter. When the layer was sufficiently thick, the circulatory flows were almost independent of the oil layer thickness. To solidify this finding, we repeated the measurements for another 2 droplets. We found a similar characteristic and confirmed that the influence of oil layer thickness on the intra-droplet active fluid flows is only effective when the layer thickness was small. Here, let's summarize our work. We found that the flows of active fluid are affected by the oil configuration, or more specifically, oil layer thickness. Circulation odor parameter is sensitive to the layer thickness when the thickness is small, and insensitive when the thickness is large. This finding indicates that active fluid flows are coupled to the oil flows near the water-oil interface, with a millimeter scale coupling length. From this indication, we hypothesize that active fluid induces oil flow close to the water-oil interface. What we mean is that when active fluid flows near the interface, its flow momentum will spread across the interface to drive oil. To test this hypothesis, we want to examine 2 points. The first is oil flow. We hypothesize that there is an oil flow, but we do not have experimental evidence. We need to check if oil flows exist. Second, if oil flows exist, they want to verify how close an oil element needs to be from the interface to experience the active fluid flows so as to be driven. These 2 questions are what we want to address to verify the hypothesis. As such, we designed another set of experiments for testing the hypothesis. Our goal is to examine the existence of oil flows. To detect the oil flow, we dope the oil with 1 micrometer sized tracers, smaller than the 3 micrometer sized tracers doped in active fluid. Then we monitor the tracers in active fluid and oil and use the particle image velocimetry algorithm to extract the flow velocities and plot the time-averaged flow field. Our plot showed that active fluid developed a circulatory flow, but in oil, there was no net flow. To confirm this finding, we plotted time-averaged azimuthal velocity across the droplet, interface, and oil. This plot showed that for a thick oil layer case, there exists a net flow in active fluid, which is a consequence of a circulatory flow. Whereas for a thin oil layer case, the net flow is nearly zero, which indicated that the flows were non-circulatory and chaotic. However, no matter the droplet was in circulatory or chaotic state, in the oil, there were no net flows. Does that mean our hypothesis was wrong? Not necessarily, because this plot is a time-averaged plot. The net flows can be zero if there is no flow, but can also be zero if there are chaotic flows whose directions cancel out on average. Thus, to find out if there is really no flow in the oil, we need to analyze flow speed. Here, we plotted flow mean speed across the active fluid, interface, and oil. In this plot, we see that no matter the droplet is in circulatory or non-circulatory states, the flow speed within a droplet is non-zero, demonstrating the existence of flows in both cases. We also notice that these flows do not stop as they cross the water-oil interface, showing that active fluid could drive the oil flow. Moreover, these oil flows died out quickly as diving into the oil bulk to a distance of approximately 1 mm, indicating a millimeter scale flow coupling length between active fluid and oil. Here, we not only verified our hypothesis, but we also got the result that is consistent with our other independent measurement of circulation odor parameter versus oil layer thickness, which also showed a millimeter scale active fluid oil coupling length. Together, we confirmed that active fluid and oil can indeed couple to each other across the water-oil interface, and that this coupling plays a critical role in the development of the intra-droplet circulatory flows. While our experiments provided a firm evidence of the active fluid-oil coupling, whether this phenomenon can be described with existing active fluid models remains unclear. To search for the finding in our experiments from existing active fluid model, as well as gain deeper insight into this finding, we attempted to establish a continuous active fluid simulation platform that can reproduce our active fluid oil coupling effect. To achieve this goal efficiently, we dived into literatures of active fluid models and found one model developed by Tong Gao from Michigan State University that meets our need. This model not only includes active fluid encapsulated in oil, but also predicted the intra-droplet circulation and water-oil coupling. So this model should be competent for modeling our active droplet system. So what is this model? The model involves fluids with multiple phases such as oil and active fluid. The active fluid in the model was driven by elongating rods, which correspond to our extensile microtubule bundles. Because of rod extension, each rod creates a pair of antiparallel forces at their ends called force dipoles. The force dipoles drive the fluid to create flows. The flow will in return move and rotate the rods. For example, if the rods are immersed in a shear flow, the rod will experience a larger drag force on one end and smaller on the other, which will result in a torque that aligns the rod with the fluid. This is called flow alignment. As such, rods create force dipoles to drive fluids, and the fluids in return rearrange the rods. Rods and fluids form a closed feedback loop to evolve side by side. We should be able to use this model to explore how the active fluid oil coupling affects intra-droplet circulatory flows. To test the capability of this model on describing our experimental results, we adapted the model to numerically calculate the time-averaged velocity fields and vorticity maps for a pair of identical droplets immersed in different oil layer thicknesses. Our calculation revealed that the droplet immersed in a thicker oil layer developed a circulatory flow, whereas when immersed in a thinner oil layer, the circulation was suppressed. We analyzed the circulation odor parameter as a function of time, which showed the similar result. For the thicker oil layer case, COP quickly grew to almost 1 and persisted for hours, whereas in the thinner oil layer case, COP fluctuated around 0. Here, we concluded that our attempt on adapting the existing model to describe our active droplet system was successful. The active fluid model captured our main experimental finding qualitatively. Next, we want to dive deeper to examine if the model can describe our system quantitatively. Here, we selected one set of experimental measurements on circulation order parameter versus oil layer thickness for this particular droplet. Then, we feed these experimental parameters into our model to calculate the corresponding circulation order parameter as a function of oil layer thickness. Comparing with the experimental results, our simulation captured a similar oil layer dependence for small oil layer thickness, where the layer thickness drastically affects the active fluid flows. We also captured the insensitivity region for large oil layer thickness. These consistencies show that our model adapted from Tonggao's work was competent in describing a 3D active droplet system. However, our model did not work perfectly. Our model predicted that further thicker oil layer did not support intra-droplet circulatory flows, which contradicted our experimental results. We have not figured out the reason for such inconsistency yet. Despite this inconsistency, our model can mostly describe our active droplet system and capture the effect of active fluid-oil coupling effect. However, we developed the simulation not just for matching our experimental data. One advantage of adapting simulations is to gain deeper insight into the mechanism that is challenging to reveal by experiments. Here, we use a simulation to examine the active fluid-oil coupling from another perspective that involved cross-correlation function. We defined a normalized same-time velocity-velocity cross-correlation function between active fluid and oil. And then we average this function over orientation, which allows us to quantify the flow correlation between a pair of active fluid element and oil element based on their separation. In other words, in our active droplet system, consider an active fluid element flowing near the water-oil interface and an oil element flowing in oil bulk. We ask how their flow velocities are correlated based on their separation. Our analysis revealed that when their separation is nearly zero, they are highly correlated. When they are further apart, they become uncorrelated gradually. The correlation function decayed exponentially. We then feed the correlation function to an exponential function, and then extract the decay length L, defined as the correlation length. Physically, correlation length means that within its length, active fluid and oil are coupled. Here we demonstrated one example. We repeated this analysis for all the simulation data for different oil layer thicknesses. And then we fit each curve to its exponential function and extracted the correlation length. Then we plotted the correlation length as a function of oil layer thickness. Our analysis showed that the correlation length first increases linearly with oil layer thickness and then reaches saturation. Let's evaluate the meaning for each of these two cases. First is the linear growth part. The linear growth indicated that the active fluid is coupled to the entire oil phase. In this scenario, anything changed in the oil is supposed to affect the active fluid directly. No wonder in our analysis of COP via oil layer thickness, COP varied rapidly for small oil layer thickness because active fluid was closely coupled to the oil all the way to the boundary. Altering the boundary shape impacts the active fluid flows. The second is a saturation regime. In this region, oil layer thickness is greater than correlation length, indicating that active fluid is only coupled to a portion of oil phase. In other words, active fluid cannot sense the oil's outer boundary. So no matter how the oil changed its shape, the active fluid flows were not affected. This corresponded to our previous analysis of COP insensitive to oil layer thickness when the thickness was sufficiently large. Moreover, the saturated correlation length represents the characteristic coupling length between flows of active fluid and oil. The coupling length of 1.4 mm is consistent with our previous independent experimental outcomes that suggested a millimeter scale coupling length. Herein, we have shown that both experiments and theories demonstrated the active fluid and oil are coupled across the water-oil interface with a millimeter scale characteristic coupling length. In other words, in the active droplet system, the intra-droplet active fluid flows are affected by the droplet environment. So, can we manually alter the droplet environment to direct the intra-droplet flows? More specifically, can we trigger or suppress the circulatory flows by manipulating the oil layer thickness? To reach this goal, we need to develop a fluidic device where we can tune the container size. To realize this goal, our first thought was to borrow the existing technology of controlling the aperture size in a camera using the so-called mechanical iris component. However, conventional mechanical iris consists of at least 6 overlapping blades. If we want to adapt the same design, then each of our blades would need to be 100 micrometer thick. Anderson tried to fabricate 100 micrometer thick blades and found that the blades were so thin that they bent spontaneously, so could not be assembled. To prevent the spontaneous bending, we had to thicken the blade. But if we thickened the blade, their combined thickness would be larger than the droplet height, which would change the droplet geometry. Therefore, to comply with our fabrication limit, while demonstrating the concept of directing intra-droplet flows from the environment, we designed our mini-fluid device with only one blade. One-blade device is not as effective as six-blade device in terms of altering the oil configuration, but at least it can demonstrate if the idea works. Here is a video demonstrating a fabricated device. Once assembled, we can manually move the blade position to alter the oil configuration around the droplet so as to influence the intra-droplet active fluid flows. Our experimental design was to first approach the blade to the droplet to a distance that could affect intra-droplet active fluid flows according to our experimental data. Then we remove the blade away from the droplet, which should allow the active fluid to develop circulatory flows. Once the circulation was developed, we had the blade approach the droplet again to suppress the circulation. If our idea works, the droplet should start with a chaotic flow, then develop a circulatory flow, and then return to a chaotic flow. Our idea was confirmed with our experimental data. The major circulation order parameter first fluctuated around 0 and then grew to 0.5-ish after the blade had stayed away from the droplet, and finally returned to plus minus 0.2 when the blade was again approached the droplet. Now, let's look at the movie to visualize the experiment. We started with the blade sitting close to the droplet. Then we move the blade away. After a while, active fluid develops circulatory flow. The process can be reversed. When active fluid was circulating, we approach the blade toward the droplet. After a while, the circulatory flow faded away. Here, we conclude the experiment was successful. The success of the experiment has two significant meanings. First, it visualized the effect of active fluid oil coupling and reassured that the environment influenced the intra-droplet active fluid flows. Second, it demonstrated the feasibility of directing the intra-droplet flows by controlling the environment without physically contacting the droplet. We can control the active fluid without touching it. It is like magic, isn't it? However, I would like to remind you that the reason we can control the intra-droplet active fluid flow using this method was because the droplet interface was fluid, so the fluids within and outside the droplet could interact. This was one of the unique characteristics of droplet systems. Another unique property of the droplets was that its shape is deformable. We had experimental data showing that active droplet developed circulatory flow in one droplet, but remained a chaotic flow in another, showing the droplet shape influences the development of intra-droplet circulatory flows. Hence, can we control the formation and deformation of the circulation by manually deforming the droplet? To achieve this goal, we need a milli-fluidic device that allows us to manually control the compression magnitude on the droplet, so we can trigger the circulation in one compression and suppress it in another. To realize such a milli-fluidic device, Anderson developed a sealing adjustable device. The device contained a movable sealing to compress the droplet at various magnitudes. To precisely control the compression magnitude, the sealing can be hung on various pre-designed platforms. Sitting on the green platform, the droplet has a height of 1 mm, blue platform 2 mm, red platform 3 mm. With this sealing movable device, we can explore the feasibility of controlling intra-droplet active fluid flows by deforming the droplet. Here is an example. In this video, the droplet was compressed to a magnitude that could support the circulation. Then we lifted the sealing to decompress the droplet, which increased the droplet height but decreased the droplet radius to conserve the droplet volume. We found that in this droplet geometry, the circulation stopped, showing that we could cease the circulation by deforming the droplet. Again, the process could be reversed. In this example, we started with a less compressed droplet where active fluid developed a chaotic flow. Then we compressed the droplet to decrease its height but increase its radius. In this thinner, wider droplet, the circulation returned, showing that we could initiate the circulation by deforming the droplet as well. Our analysis on evolution of circulation order parameter agreed with our observation. The decompression stopped the circulation, the compression triggered the circulation. We could trigger or suppress the circulation by deforming the droplet. The success of this experiment may also provide insight to cellular biology. One of the important intracellular activities is that cells will develop a cell-wide flow to promote nutrient transport called cytoplasmic streaming. The cytoplasmic streaming was known to be driven by molecular motors similar to our active fluid system. So formation and deformation of the cytoplasmic streaming may be controlled by deforming the cell. Deforming the cell to a shape may promote the cytoplasmic streaming, whereas deforming to another shape may suppress it. Our finding may pave the path to developing a new cellular treatment to control the intracellular activity without invading its interior. To sum up this talk, we demonstrated that confining active fluid in a fixed compressed water-in-oil droplet could support the development of intra-droplet circulatory flows. Our extensive data showed that the circulatory flows depended on the oil configuration, or more specifically, the layer thickness of oil that surrounded the droplet. The reason for such dependence is because active fluid could induce a thin layer of oil flow across the water-oil interface. The layer thickness suggests that the flows of active fluid and oil are coupled with a millimeter-scale coupling length. Then we established a continuous active fluid simulation platform that reproduces experiments and suggests an active fluid oil coupling length of 1.4 mm, which is consistent with our experimental data. This consistency suggests that our simulation can be used to guide experimental design of active droplet system. We then demonstrated two examples of applying this new finding to milli-fluidic technology. One example used mechanical iris-based design to control the circulatory flows in a droplet without touching the droplet. The other used a sealed movable milli-fluid device that could control the compression magnitudes on the droplet so as to trigger and suppress the circulation in real time. These examples demonstrate that the more we know about the active fluid and active droplet system, the more technology we can develop to utilize the energy in the active fluid to accomplish a task that we could not accomplish before. The above is a talk I would like to share with you about the recent progress in my lab. Here, I conclude the talk and thank you for your attention.